didn't do super well on the first test, what do I do? Well, probably what you need to do is take a second and reflect. Why didn't you get the grade that you wanted on the test? And please don't tell me it's because we tried to make it hard. I have to tell you that that was one of the more straightforward tests that I've seen. So, and the class average was not awesome. It's okay. It's okay. So many of you did all right, but some of you clearly struggled. We get it. This class is hard. Lots of vocabulary words, lots of things you need to learn. Some of you haven't had bio since 9th grade, and that was a very long time ago. But if you did not get the grade that you were hoping for on the first test, statistically, the grade on the first test is your grade, your final grade in the class. Okay? That's not 100% true, but that's often true. Right? Secondly, there's the course material for this class is cumulative. So the final exam is not cumulative. We're not going to ask you a question from chapter 1, okay? But the information is cumulative. So today, so far, several times, I have called attention to information that you learned earlier in the semester. So if you didn't get the grade that you were hoping for on test 1, then you may not know the material for test 1 as well as you would like, and maybe not as well as I need you to in order to actually fully understand everything that I'm teaching you. So, if you did not do as well on test 1 as you would have liked, reflect on what you did and didn't do and what you could do differently. Remember that I said on the first day of class, this course meets the national recommendations for how these types of courses should be set up. So that is 2 to 3 hours of homework that you come up with for yourself for every 1 hour of lecture. This is a 3-hour lecture, okay? So it should be 6 to 9 hours of studying on your own at home, plus 1 to 2 hours for the lab. Now, if you don't know what to do with 6 to 9 hours of how do you study bio, well, we give you sample test questions, both multiple choice questions and short answers. We give you vocabulary words. We give you the PowerPoints. So go to the PowerPoint. If you cannot use your vocabulary words to describe what is happening in the pictures to your classmate, you do not know the information as well as you need to. The tests are multiple choice, but they are designed so that you should be able to read what is called the STEM, the actual language of the question, come up with the answer, and then find the answer in those options. Answer options. And so to do that effectively, you really need to know the material. Sure, you can do testing strategies and, use the process of elimination and whatever, but generally you know the answer or you don't know the answer. Many of you have come to my office and they're like, well, I'm studying for the final. You shouldn't be studying for the final. You should be reviewing for the final. Every week you should be studying. Every week you should have a clear set of goals. And the goal shouldn't be, I'm going to spend 3 hours studying. The goal should be, I'm going to understand these pages in the textbook. I'm going to be able to understand them well enough that I can use all the vocabulary words on the list and I can use them clearly and I can explain it to my mother or my classmate or whoever the poor sucker is that's stuck beside me on the bus, okay? So, if you're not sure if you're studying right, come to my office hours. Now, I know I only started lecturing to you guys today, but so far no students have come to my office hours, and I can tell you that the probability that anyone will come to my office hours except the day before the exam is very low. So please use my time, right? I'm the one teaching you. I was a student once, and maybe my student life experience isn't the same as your student life experience, but English is my second language, and I'm dyslexic. So I certainly have some understandings of the struggles that you went through as a student. I did have the equivalent of AP Bio before I was a student in the class, so that was a little different. But my class had 2,500 students in it. You guys have 300. So my teacher was about this big. I was on the third balcony. And we had overheads, if you even know what those things are. So the teacher would write notes, and we'd have to copy them. So please come and see me. I can help you figure out how to study. I can help you figure out what it is that you can try to do to really improve your grade moving forward. We have certainly had students who failed the first test and gotten a 90% on the final. So while for most students the grade on their first test is their final grade in the class, there are lots of exceptions, and maybe that exception this semester will be you. But it can only be you if you are proactive and figure out what can you do better. Did you go to the tutoring center? We have free tutoring, and I train them every week. They know what I need you guys to know. These are not just Joe Schmo off the street. They've been working with me for several years. There's Morales' office, Dr. Tajirian's office hours, your lab instructor's office hours, etc. Again, as a part of the contingency plan, everyone will be moving their office hours to an online Google Hangout. So if any of you have never logged into your Queens College Google account, please do so. I'll send you an email with instructions for how to do that. You all have a Google account through the education site. And then you can participate in online office hours if we end up not being allowed to meet face-to-face. Okay, so I don't want to keep you here too late, and I'm already running a little late. So again, thank you for your patience. The next step of what we're going to talk about is gene regulation. So the idea is really simple, right? We've just finished talking about how you go about making a protein. But I started off today by saying that each of you are who you are because of the unique sets of proteins that you are made of. And so we want to think about, when are certain proteins made? How does the body know which ones to make, which cells to make them in? So your heart is going to need a different set of proteins than your skin. And you're going to need different proteins in different phases of your life. While you're growing, you may need one set of proteins. While you're sleeping, you may need a different set of proteins. And so regulating how and when genes are turned on and off in both development, when you're growing, and as an adult is really important, because you want to sometimes be able to turn things on quickly, sometimes turn things off quickly. Sometimes you want 30 things to happen at exactly the same time. So you need a way to regulate this process. And so that's what we're going to talk about next. There's a section at the end of this which I may skip because we may be short on time. Anything that I skip, if I don't teach it the next class, it's not on your test. Okay, so the lectures are set up so that everything I really want to make sure that you have in order to be successful moving forward in your major or writing your MCAT or whatever is the first 2/3. And if I cover the last bit, awesome, because it's fun facts. If I don't, I won't put test questions on the test for it. So don't freak out. If I cover it, it's on the test. If I don't, it's okay. So, and thank you for staying. I really appreciate an audience. I'm assuming it's this empty because I'm theoretically posting this online. Hopefully it works. Okay, so how do genes become regulated? For most of today, we're going to talk about gene regulation in prokaryotes, partly because it's easier, partly because it was discovered first, and mostly because that's the examples the textbook give. And if I give you a whole bunch of stuff that's not in the textbook, you're all going to go home and go, oh my God, what did she do? So we'll wait for that until you're a little further along in your major. Now, eukaryotes do all of these same things too. They just have some added layers of options because they have a nucleus, because they have introns, because they have a poly-A tail and a G-cap. They can regulate things a little bit differently. So this is all basically happening, in this case, in prokaryotes. Eukaryotes have more options. So, if you just take a second and think about it, if we want to change how much protein is present, every cell has the same amount of DNA. In theory, they all have the same amount of enzymes and materials for making mRNA and for making proteins. So what kinds of things could you do so that you have less protein in a given cell type? What could you do? Yep. So you can modify the expression of a particular gene. If you were to modify the expression of a particular gene, what would be the result at the next step? The messenger RNA, right? You could change how much messenger RNA there is, right? You could have lots of messenger RNA. You can have a little bit of messenger RNA, right? Now, what about about at the next step? Can you regulate how much proteins are made from the messenger RNA? Maybe, right? Cells are smart. So maybe the cell has a way of saying, take that messenger RNA and store it away right now, so that, because we want it available, but we're not going to use it right now, right? So, and then the other piece that we have control over is how long the messenger RNA is present in the cell. If the messenger RNA is around for a long time, then you're able to make protein for a long time. If it's only around for a short period of time, then you can only make protein for that short period of time. Proteins can be regulated the same way. Cells have complicated systems for chewing up proteins, degrading them, and some are degraded quickly, some are degraded slowly, and some are degraded only in response to some environmental change, some condition, etc. Okay? So depending on the circumstance, the cells can respond and have the right amount of protein at the right time at the right place. The last thing that we can do is we can prevent the function of the protein. So you guys learned a little bit about this earlier in the semester when you talked about inhibitors. So there's competitive inhibitors, non-competitive inhibitors, and so if you end up with a lot of that protein, you can still change the function of that protein depending on whether the inhibitor is present. So when we're regulating how much messenger RNA is made, we can regulate that process in two directions. We can have a negative regulator. Do you think a negative regulator would make more messenger RNA or less? Less, right? So what I'm trying to show you, and I was telling this to some people up front, is this class has a lot of stuff to memorize, but there are also ways you can learn it by thinking about it. So if you think about, oh, the word negative regulator, that probably means negative or less, right? What about a positive regulator? It's going to go more, right? You're going to go up. So take advantage of that language and think about what you're saying as you go forward. So within the promoter, remember we talked an hour ago about how the gene will have a promoter, and it's the promoter that starts where the messenger RNA is made. That promoter can have negative regulators or positive regulators, and that will change how much messenger RNA is made. And so here is an example of a negative regulator. So what you'll see is this DNA right here, and here is where transcription would start. So here's the transcription start site, and this is inside that promoter region. So in that promoter region, you can have a repressor binding site, and a repressor is going to be a negative regulator. So if that repressor is present, then the messenger RNA is not made. Okay? Now, if you have at that site an activator binding site, then an activator protein, in this case shown here in green, that activator protein can bind the promoter, that binding site, and promote messenger RNA synthesis. Right? So if there's no regulators present, no positive and no negative regulators, then usually they'll make like a little tiny bit of messenger RNA. If the negative regulator is present, it's closed, it's off. If the positive regulator is present, it's on. This is also an oversimplification. Next class, we're going to look at circumstances where you might have 6 different positive regulators and 4 different negative regulators, and they work in combination. And that can allow for greater regulation. There was a question. You still have a question?
Yeah, I was just wondering, can it be you said if there are repressor proteins bound to the repressor binding site, then it's cut off. But if there is none, they sort of act like a positive?
Right. So that's a really good question. If there is a negative repressor binding site, but that protein is not present, what happens? If there's no positive signal, if there's no negative signal. So most genes will have what they call a basal level. So most genes will just make like a little bit of stuff unless they're told make more or told shut off. At the end of the lecture today that I need to make sure to cover, I'm going to talk about some strategies about how the DNA is packaged that can turn things off permanently. So we do have ways in our body to just shut off the DNA without having the repressors present. But in most cases, just make a little bit. Okay? Question? All right.
You said that there can be more than one repressor activated or present on the promoter.
Binding site, yes.
So, like, what would be like
Well, so when we talk next class, we're going to look at genes that are involved in regulating how an embryo grows. And what you're going to see is that in those cases, there'll be a gene that's responsible for making different parts of your body. And it's done in a concentration-dependent fashion. So if you have a tiny bit, then you make the top of your head. And if you have a little bit, you make the middle of your head. And if you have a lot, you make the bottom of your head. And so the exact amount of messenger RNA and protein that is made is controlled as a ladder, steps. And so you have lots of steps that you can make, yeah?
So these, like, multiple activators and repressors would be present in, like, stem cells?
So they're always present in the DNA, but how many are present in the cell that turn on or off a particular protein is going to depend on the cell, where it is in the body, where it is in the growth phase, where it is in the time of day, all of those things. Every cell will be a little bit different. You had a question too?
So where exactly is the promoter located?
So the promoter in this case is actually probably a whole chunk of material in front of that start site. So this is the start site, and that green section is the promoter, and then it keeps going. Okay, so there's going to be, they always oversimplify it, right? They're just showing you, in the simplest of case, this little sequence of nucleotides is the repressor site, but it could be much bigger. It just keeps going off to this way, yeah. Okay, and then remember, the left versus right is a standardization in which they draw things. So in this case, the DNA, the gene is the 5' to 3' on the top. The template is the 3' to 5' on the bottom. And then the messenger RNA is made from that. But that's a drawing convention. The cell does not care which direction it points. It could just as easily be flipped the other way. Okay. Here are a bunch of words. Basically, these words say that in a cell, if you are a certain kind of E. coli, which is a kind of bacteria which are in our bellies, that you want glucose as your food source. Glucose is yummy, right? That's sugar, your table sugar. It's yummy, right? Sometimes there's lots of glucose in your environment and you want all the proteins necessary to eat glucose. But sometimes life sucks and there is no more glucose and all you have is lactose. Now you want to eat that because it's better than starving. So when there is no glucose, but there is lactose, then we want to turn on all the genes that let us use lactose as a food source. In these E. coli bacteria, so remember these are prokaryotes, there are what are called, there are 3 proteins necessary to be able to use lactose as a food source, and they are listed here. So there's beta-galactosidase permease. Now again, listen to the words. Permease means permeable, and it's an enzyme-y thing. So this is a thing that lets lactose into the cell. And then there is a beta-galactosidase which hydrolyzes the lactose, which is a fancy way of saying it starts to digest it. And then there's a beta-galactosidase transacetylase, and that adds this acetyl-CoA to the sugar, which probably helps it get into the citric acid cycle a little bit easier, because if you remember, acetyl-CoA is part of the citric acid cycle. But these are 3 proteins that we know are important in order to digest lactose. So we want to make those, but we only want to make those when there's no glucose, because if there's glucose, we want the glucose. And when there is lactose present, because we don't care about making them if there's no lactose. So the cell has a way of saying, is there lactose present and is there glucose present? What do I need to be making in order to be able to eat? It turns out that there's a small delay in when you activate these genes. So if you have no glucose present and you want to start eating the lactose because it is present, it takes a little bit of time. And it takes a little bit of time because the cells need to make the messenger RNA, and then they need to make the protein, and then they can start digesting the lactose. And that's what's shown here. So there's an inducer that says lactose is present. It's time to start making the genes to make the messenger, try to activate the genes to make the messenger RNA to make the protein. And this takes a few minutes. So we add the inducer. In this case, the inducer is lactose. Oh, look, lactose is present. We can start eating that. So we're going to have a delay between when we can do that, when we have that inducer present and the amount of beta-galactosidase. So this is beta-galactosidase over time. So the beta-galactosidase, that protein increases in volume in the cell, even when that inducer is removed. All right, so we add lactose, we say let's make these proteins, we make the proteins, we remove the lactose, and we still make proteins for a little while. And this is if we look at the RNA, we can see why. So here's again over time, and this is the amount of RNA for these 3 different proteins. So we can say the inducer is added, we start to make the messenger RNA. As we're making the messenger RNA, we're making that protein. Now the inducer is removed. When the inducer is removed, we stop making messenger RNA. But there's still messenger RNA in the cell, so we continue to make protein for a little while. So this is a generic way in which many metabolic pathways are regulated, and that's what's shown here. So this first run looks very similar to the very first experiment that we looked at today, right? So we have a precursor, enzyme 1, which is made by gene 1, converts the precursor to product A, and then product A is converted to product B by enzyme 2, et cetera, until we get our end product. Now, sometimes we want to stop making these things along the way, right? If we have tons of the end product, we don't want to put a lot of energy into making the stuff in the middle. And so we can feed back and inhibit the process. So that's what's shown here. So this end product is going to inhibit the ability of gene 5 to be made. If we don't make gene 5, then we don't make enzyme 5. Similarly, it's going to inhibit all these other genes. And this can be done at the level of transcriptional regulation. Okay, so again, we're transcribing. So what this is telling us is that the end product can prevent the messenger RNA synthesis. The end product, in this case, can also regulate the enzyme's function. So in this case, the end product is regulating allosteric regulation, changing the shape of the protein, and keeping enzyme 1 from working. So these are 2 different ways in which a metabolic pathway can say, I have enough stuff at the end, I'm going to go backwards and I'm going to turn it off. And pretty much, we're going to see through the semester, almost every single biological system works this way. The end product, when there's too much of it, is going to go back and say, enough. Oddly, the only thing that doesn't work that way are sex drive hormones, which positively feedback. They're like, oh, I got a little, oh, I'm going to have a little more. And then they crash and say enough, and they stop. But they're the only ones that we talk about in this class where there's a positive response to that, that end product. Okay. Correct. This one is regulating how much messenger RNA is made and therefore how much protein is made. This one is regulating the activity of that enzyme. So if you make lots of enzyme but it's all inhibited, then you are not going to have that activity. Okay, question?
So why would you both how much mRNA is made and
Good question. Why would you do both, right? Why wouldn't you just pick one? So there's different systems will maybe use exclusively one or exclusively the other, or they will use both. And it depends on how important it is that the system is turned off quickly. So you can imagine scenarios where it doesn't really matter if there's a little extra and it's waiting around, and other scenarios where you're like, oh crap, I better stop right now. And so this allows you to determine how important something is or not. Okay. Yeah?
Can this be applied to larger metabolic pathways like nitrogen cycles?
So this can be applied to pretty much every biological process. So it can be applied to metabolic systems, it can be applied to hormonal systems, it can even be applied to neural networks. So when we look at how the brain functions, we have a similar feedback loop where some signal at the end can feed back. So this is a very generic concept, absolutely. Okay. So this ability for E. coli to use lactose as a food source is because E. coli have what is referred to as the lac operon. So the lac operon is kind of a weird phenomena of prokaryotes. So in eukaryotes, we have one promoter and one messenger RNA, one gene, pretty much all the time. In prokaryotes, they do this funky thing where they put a bunch of messenger RNAs that are all involved in the same biological process right next to each other, and they control them with one promoter. Okay? So that's what this is. So if you look here, let me make this bigger. You'll see this section right here of this piece of DNA is referred to as the lac operon. Now, these blue pieces here, beta-galactosidase gene, beta-galactosidase permease gene, and beta-galactosidase transacetylase genes, these are the three sequences that become messenger RNA and ultimately protein. They are regulated by the same promoter region. So here's the lac operon promoter. This is referred to as the operator. It is not going to make messenger RNA, so some people include the operator as a part of the promoter because it's regulating when a gene is turned on and off. So when you have the right environment to be making or digesting lactose as a food source, you're going to make all three of these messenger RNAs, and you're going to regulate that as one cassette, one module. So you turn the whole thing on. Interestingly, there are a couple of other proteins that we need to regulate this process. One of them is called the I gene. Okay? And the I gene turns out to be right next to the lac operon. So what you can see here is this is the promoter for the I gene, and this is the actual messenger RNA to make the I gene. What does the I gene do? Well, the I gene is the repressor protein that represses the lac operon. So the lac operon is going to be off as long as this I protein is able to bind. Okay. So, somewhat unique feature of bacteria, of prokaryotes, are having these operons, multiple genes together, with a single controlling promoter. Now, yeah?
So, the gene I promoter is the one we covered before, turning off the black operon.
Yes.
And it's only done when there's a lot of...
We're going to show you each of those examples. So, bear with me. We'll get there. Okay? So, in this case, we have a system that is regulated on multiple levels, right? We want an inducer and we want a repressor. We want the system off anytime glucose is present, and we want the system to be able to turn on anytime lactose is present, but only on when glucose is absent. So no glucose, yes lactose, we make these genes into messenger RNA and then protein. So we have both a need for induction, for positive regulation, and for repression. In this case, we have one more layer of regulation, and that's called an activator. So we also want to say, go. So let's look at each of these steps. So the first thing is, in the absence of lactose, we have the repressor present, and it's bound to the operator of the lac operon. And remember, this repressor happens to be made by the I gene, which is right next to it, but it doesn't need to be. It could be anywhere else. And so this repressor is active, it binds to the operator, and it prevents transcription. So we're just off. And this is going to be a circumstance where glucose, right, so there's no glucose, and there's lots of glucose, there's no lactose. All right, so we want to be able to get rid of this repressor. So we want to get rid of this repressor, in this case, when lactose is present. Because when lactose is present, we want to be able to turn the system on. Now, we're not going to turn it on unless glucose is off, or glucose is absent, but we want to be able to activate it So in this case, what's nice is that the system is dependent on its final product. So here's that repressor. Because lactose is present, the repressor becomes inactive, right? And now we know lactose is present, the repressor is active, we can make the messenger RNA necessary to make the proteins. Now, we can make this, and here they all are, but we don't make a lot of it. We just make a little bit. Because we don't have an activator. We aren't saying go. And so the next step that we need is to be able to have that go factor. And the go factor, again, is going to be dependent on the fact that glucose is now not present, right? So when glucose is at low levels, we have an activator. This is the activator. It's called CRP. And CRP, in the presence of low glucose, is going to bind cyclic AMP, and cyclic AMP will then bind the promoter and will act as an activator. And so now, instead of making just a little bit of the messenger RNA, we're going to make a lot of the messenger RNA. So we have these two parallel strategies for regulating this process. When glucose is present at high levels and lactose is present at high levels, there's not going to be any repressor bound, but we don't want activation of this messenger RNA, and so that's what happens here. So there's no repressor, but there's also no activator, and so we don't really make any of these products. And again, that's because we want to use the glucose, so as long as the glucose is present, we can do that. So within, again, this is now a eukaryote, right? Because we can see the edge of the nucleus. We have a set of places where we can regulate this, similar to what we saw in the prokaryotes. We can regulate it at the level of the chromosomes. So you learned during meiosis and mitosis that the chromosomes wrap around histones and they can be tightly packed, right? That also means sometimes they're loosely packed. So when the chromosomes are loosely packed, enzymes can get in there, transcription can occur. When they're tightly packed, it's very difficult. We can also regulate the actual synthesis of the messenger RNA, and we can regulate the transport of the messenger RNA. If the messenger RNA is in the nucleus, it's not getting made into protein, right? So if we prevent it from leaving the nucleus, we can prevent it from making protein. Now, we can also, in eukaryotes, regulate what happens to that messenger RNA. So is that messenger RNA going to be inactive and degraded? Is that messenger RNA going to be translated into protein? If it's translated into protein, is that protein going to be activated? Or is that protein going to be degraded? So several places along the way that we can regulate this synthesis of proteins. So again, comparison of prokaryotes and eukaryotes. In prokaryotes, the genes are going to be located in these clusters called operons, and these clusters tend to regulate genes of the same basic function, whereas in eukaryotes, they're usually quite distant from each other. They have their own promoters. They're regulated independently. Prokaryotes have only one RNA polymerase. Eukaryotes have three. You don't have to know which one does what, but there's three, one for making tRNAs, one for making rRNAs, and one for making messenger RNA. Because you have these, in prokaryotes, you have lots of genes all together with one promoter. You don't have a lot of promoters, whereas in eukaryotes, you have lots of promoters. And then in terms of initiating transcription, in prokaryotes, the RNA polymerase is going to bind right to the promoter. And eukaryotes, you're going to have lots of different things that are necessary to come together to properly activate a gene. So what does that look like then in a eukaryote? So in a eukaryote, if we look at the beginning of where a gene is, this is your entire gene. This is the coding sequence. This is the part that makes the messenger RNA. And then this is the promoter. The promoter almost always contains what is called a TATA box, and it's called a TATA box because the sequence inside it is TA. We're very creative sometimes. And a TATA box is going to be the place that helps this whole transcription start, and it's going to be bound by an enzyme, a protein called TF2D, and TF2D is the TATA binding protein. And it's going to sit in that spot, but it's not going to say make the messenger RNA until additional factors come in and help. So there's a transcription factor, transcription factor B, for example, might come in. And there may be a lot of other transcription factors that are necessary in order for the protein to be synthesized. And that can be really important, especially in embryonic development, because you put all these different signals together and you turn on a specific set of genes to make the cell type that you're interested in. And so only when all this TF2D and this generic transcription factor and a host of specific transcription factors are sitting and waiting in the right place can the RNA polymerase come in and make the messenger RNA. So again, lots of steps for being able to regulate when DNA is translated into or transcribed into messenger RNA. Another really cool thing about DNA is that it's floppy. And in eukaryotes, we have lots of DNA. And sometimes a regulatory element for saying when a gene should be turned on or off may be a long way away from the promoter if you just look at it in a linear fashion. But if you look at how the DNA loops back on itself, it actually can be very physically close. So if my two hands are far away, if I loop them, I can bring them together. And so we can see that here. So these are referred to as enhancers. So here's an enhancer that can be quite far away, like 2,000 nucleotides away. But when the DNA loops, it comes really close together. And so this is a feature of eukaryotes, not prokaryotes. And if you take genetics, you're going to learn a lot more about these types of enhancers. And they are called enhancers because exactly that, they enhance the efficiency of making the messenger RNA. All right, so we talked about a whole bunch of things that bind DNA. I like structure-function relationships. So how are things binding DNA? What does that mean? What does that look like? So we all know what the basic structure of DNA is, right? It's this helix ladder that spins, and it's twisted. And we have some idea of what protein shapes are. I suspect you talked about beta-pleated sheets and alpha helices. And so if you look at the structure of a protein, proteins that bind DNA have specific structural domains. They have specific shapes. These are called structural motifs. And this is referred to as a helix turn helix or a helix loop helix. So this is an alpha helix and a turn and an alpha helix. And you often find these in proteins that are able to bind DNA. And we can see that here, right? So here, the helix turn helix is sitting in the major groove, that big gap in that DNA molecule. And this is what that looks like for real. So this is actually that Lac repressor, that I gene. And here's the majority of that protein, that Lac repressor protein. And here is the helix turn helix motif interacting with the major groove of the DNA. This is a dimer, so there's another one on the other side as well. And these are sequence-specific interactions. So the protein, those helix turn helixes, has a protein organization that is able to detect the nucleotide sequence. And so it's not randomly binding the DNA, it's finding a match. And that's important because that's how you know, okay, I'm at the repressor site in the protein, or I'm in the activator site, I am in the activator site of the DNA, and I should make the messenger RNA and thus the protein. There's another common shape that DNA binding proteins have. They're called leucine zippers, and they're called leucine zippers because leucine's amino acid. That amino acid is found in this little row down the center, and these two proteins look like they zipper together where these leucines are located. And then they have these little feet which interact again with the major groove of the DNA, and that allows them to be able to tell what the DNA sequence is, find the right binding site, and either activate or repress the messenger RNA synthesis. So the activator proteins and the repressor proteins have a specific three-dimensional shape. that let them bind to a particular sequence in the DNA. So if the DNA is TA, like we saw in the Tata box, then the protein that binds that Tata box has a particular shape that lets it say, this is the Tata box, this is where I should bind. Okay, so it's the shape of the protein that lets this work. I feel like we all need to do the 7-minute stretch. If you see a lot of little sleepy faces. Okay. So, it's the presence of these transcription factors that determine whether a protein is made or not, that determine whether a messenger RNA is synthesized or not. So, when an embryo is growing, we have lots of different types of cells. One type of cell that we might make is a neuron. In order to make that cell a neuron, there are transcription factors that say to this cell, you are a neuron. Turn on the genes that makes the messenger RNA, that make the protein, that allow you to function like a neuron. If we can figure out which those genes are, what the signal is that says you are a neuron, then we have a lot of therapeutic potential, because we could turn those genes on in another cell type and change one cell into another cell. So we could take some skin from an individual, convert those cells into cells that will make neurons, inject them into the brains of patients with Alzheimer's or dementia, and in the right circumstance, help heal those brains. That sounds really far-fetched, but I can tell you that we're already able to do that with certain cell types. We are able to repair vision in some people. We're able to grow skin for people. We're almost able to grow blood vessels and even some heart tissue for people. They have successfully grown a bladder in a dish. They have yet to implant it in a person, but we're getting there. Certainly in your lifetime, we'll be able to take some cells from you and grow an organ for you, and the need for transplants from other places will be much less. Okay, so how is this controlled? So sometimes we need a master regulator. So again, in the example of embryonic development, if I want a whole bunch of, if I want a cell to become a muscle cell, I need 600 genes turned on all at the same time. Not just one, because I need ones that make all the different parts of the muscle proteins and all the different shapes of things necessary to make a functioning muscle cell. So cells have kind of a master regulatory system that by turning on this one transcription factor, now 100 genes will be made, and those 100 genes will quickly make or activate the synthesis of another 100 genes. Similarly, if there's a stressful situation, like if a cell gets really hot, then it needs to create a whole bunch of proteins all at the same time to help protect the cell. And so that's kind of the example that's shown here. So here what we have is a little piece of DNA. This DNA is going to make one of these master regulators. So we have a stress factor, maybe there's an injury, maybe it's temperature, and that stress factor activates the synthesis of this one gene. Now this one gene makes a protein, lots of this protein, and this protein is going to go and turn on gene A, gene B, gene C, maybe 100 different genes, and each of these genes then are going to allow the body to very quickly have a complex response to a very simple stimulus. Again, temperature, decision to be heart cell, etc. Question?
So the master regulator is that little top gene, that's that top gene in the top?
That's the protein made by that top gene is the sort of master regulator, yeah. And that's my word, that's not a word that like other scientists use, but I find it helpful for you guys to understand what I'm talking about. Okay, so it's a transcription factor that's going to turn on a lot of genes all at the same time. And the example that's shown here is this thing called a stress response element. So any gene that has a stress response element will be turned on when the stress response protein is made in response to stress. So stress will make the messenger RNA be produced for this stress response protein, which then binds to the stress response element and makes a bunch of other proteins. Yep?
So after the genes are turned on, what do they make?
So each of those are going to make a messenger RNA, which then makes a protein. What it makes depends on the circumstance. So in the case of temperature response stress, it turns out that temperature is really going to screw up the three-dimensional shape of proteins. So there's a whole bunch of proteins called chaperone proteins, and they act just like you might imagine a chaperone, and they keep the boy and the girl from touching too much on their date. So they hold all of the proteins in the right shape. They act as a chaperone and make sure that the proteins are able to behave properly. So that's one example of a protein that might be made in response to a heat stress. You may also need to make 15 other types of heat response proteins. Right? So it depends on what the source is and the cause of that response. All right, so viruses. So the reason we're talking about viruses is they have some really interesting ways of regulating their DNA, and because they're topical for today and this week. So viruses are generally not referred to as living, okay, because they're parasites, they need another organism in order to be able to replicate. They're often referred to as particles. So a virus particle can be referred to as a viron, and they are acellular. They are usually composed of only nucleic acid. And I say nucleic acid because there's RNA viruses and DNA viruses. They're usually only composed of the nucleic acid and maybe a few proteins. The virus is going to use that host set of cells to carry out whatever metabolic functions it needs, make lots of protein, make more nucleic acids, etc. And we can study them to really understand how genes are regulated because they don't really have very much DNA. These weird little moonlander alien things are what are called bacteriophage. Phage is another word for a virus. So a bacteriophage is a bacteriovirus. Dr. John Dennehy on our campus actually studies the relationship between the evolution of the phage and the bacteria. And so here's an example of one of these bacteriophage on a bacteria, and you can see that the virus is injecting its genetic material into the cell, and you can see that the cell is starting to make a bunch of these viral particles inside the cell. Now, viruses have two ways of living. They can live inside the cell and just kind of chill, make some copies of themselves, bud off, and release a few copies of the virus that can go elsewhere. Or they can make a ton of copies of themselves and make the bacteria that they're inside explode. Okay? Same thing is true in humans. So the influenza, the flu, it can live in your cells and just kind of chill for a while, produce more virus, which then gets released and other cells will get it, or it can just build up in really huge quantities in a particular cell, make that cell explode. And so when a virus is really happy, it makes cells explode. That's called the lytic cycle. So the lytic cycle is what's shown here. So here we have a bacteriophage binding to a bacterial surface, and in the response to getting into the cell, the first thing that it's going to do is activate these early response genes. It's going to use the bacterial replication system, because it doesn't have its own polymerase. It's going to use the bacterial polymerase, and it's going to make these early sets of genes. And these early sets of genes then are going to be transcription factors that go on and activate. So they're going to make some proteins for the cell, but mostly they're going to make transcription factors that activate the late genes. So then we'll make more of the genetic material for the virus, and then we're going to make a whole bunch of the virus proteins. By making the virus proteins, we can make the virus shape, the little moonlander. We can stuff the genetic material inside the little moonlander. And then when we make enough of them, we make the cell explode. The cell dies, but it doesn't care, and the virus doesn't care. It goes and it infects a whole host of other cells. Yes?
So when the virus injects its DNA into the host, can't the host pick up that something that's not supposed to be there is there?
Good question. So bacteria have kind of the equivalent of an immune system in that they have a strategy for preventing the infection. So they do several things to prevent the virus from binding. They do other things to prevent the virus from injecting the DNA. The most common strategy is that the viral DNA is chopped up into small pieces. And I think you guys have you done the PGLO lab? So in lab class, you're going to do DNA transformations where you put plasmid inside bacteria, and then the bacteria fluoresce green. Have you done that one? Some of you have done that one. Okay. So that experiment depends on the ability for us to chop the DNA into little pieces. We do that with the restriction enzyme. Restriction enzymes are enzymes that are normally present in bacteria, and their job is to cut up the DNA of foreign invading viruses. So as the virus is injected into the bacteria, it can be recognized as foreign and cut into small pieces. But sometimes there's so many virus, or the sequence of the virus has changed, and so that it's not recognized as foreign. But yes, you can do that. Okay, so that's the lytic cycle. That's how cells explode. There's also what we call the lysogenic cycle. That's when the virus just kind of hides inside the cell and chills out. And so depending on the circumstance, you're going to either do what we just saw here, which is the lytic cycle, make them explode, or do the lysogenic cycle. Yes, there are lots of labels on this. I'm not testing you on any of the individual steps. What I said is what you need to know. So how does a cell pick between the lysogenic and the lytic cycle? They're going to be completely different genes that are turned on and off. And right now, that's what we're focusing on. Which genes are turned on and how. And so that's what we see here. So the bacteriophage, the virus, is going to determine whether it's going to be lysogenic or undergo lysis. So it's either going to explode or chill, depending on how happy the cell is. So let's go through this really in a stepwise fashion. So here is the genetic material of the virus. And here we have a gene called CRO. So I'm going to zoom in on that because I want to make sure you can see it. So there's this CRO gene. Here's the CRO promoter. So the CRO promoter is going to go in this direction and regulate the CRO gene. And then this is called the CI promoter, and the CI promoter goes in this direction, right? So we talked about how we tend to draw everything going the way we read. Well, in this case, one goes one direction, one goes the other direction, okay? And the CRO gene, when it's present, is a positive regulator for itself. It makes more of itself. And it also turns on a series of lysis genes. So lysis, again, means explode. So when the cell's ready to explode, the virus is going to generate material through these lysis genes. Now, the CRO protein is going to inhibit the CI protein, or C1 protein, and C1 is important for lysogenesis, right? So you can see that if you have lots of CRO protein, then you're going to turn off, you're repressing the C1 genes, and you're going to turn on the explode lysis proteins, right? So the question then becomes, how does a cell know it's going to make lots of CRO or little CRO? So if it makes lots of CRO, then the cell's going to head into lysis and explode. If it makes a little bit of CRO, then we can go into the other direction. So C1 and CRO are going to be made when there's an early infection. So as soon as you get an infection, you're going to make a little of both. And it's really a first come, first serve, right? Sometimes you make a lot, sometimes you make a little. Just kind of shakes out randomly. But if the host cell, that bacteria, is dividing rapidly, then the CRO synthesis is low, and that allows the C1 protein to accumulate. And if the C1 protein accumulates, then you enter this lysogenic phase and you don't explode. Now this actually makes some sense. So if a cell is growing quickly, it's likely to divide. It's likely to be a happy environment for the virus to sit in. So now it can sit in the bacteria, and the bacteria will divide, make lots of copies of itself, then the virus doesn't have to do any work. And then when the environment becomes stressful, and the bacteria is not dividing rapidly anymore, now it can make lots of copies of itself and make the bacteria explode, and then go and find a new set of bacteria to grow in. Okay, so this is again an example of both positive and negative regulation, depending on the circumstance. Now let me just, right. I have like 15 minutes, right, 9:30, because we took our break? Okay. Okay, so couple then examples of how we regulate genes. Another example of how we regulate genes is called epigenetics. Now, epigenetics is a new and big, you had a question?
For the slide, for the virus, does virulence affect which gene?
Very good question. So virulence, for those of you who are not quite sure, has to do with how likely a bacteria is going to make you sick, et cetera. And so it's very much dependent on how the virus works and what the host is. So different viruses under different circumstances will produce different numbers of viruses. So, for example, I'm trying to think of the actual numbers. All right, these are not accurate numbers because I can't remember the actual accurate numbers. Some bacteria, when they undergo lysis, will only release 100 viral particles, whereas another virus might release 1 million particles. And 1 million is actually not an unreasonable number for these. They can create a ridiculous number of these viral particles. And so one way that virulence is determined is by how many viral particles are generated. The second is how quickly the cell will burst. So if you think about what makes you sick, one of them is how many cells are infected, and the other is how many of those cells have died and exploded, right? So, so it's in the case of us, it's a complicated question. There's another question, yeah? So the question is, can the virulence depend on the host situation? And the answer is absolutely. So there are a couple of ways in which how sick you get depend on who you are. Some of it is totally out of your control. So a virus has to find a binding partner on the surface of your cells. So some people have different surface proteins and they may be more susceptible or less susceptible. Some viruses only bind to one cell type. They might only bind to a neuron. Or if you've ever had, like, sometimes you'll get a really weird flu where, like, this muscle hurts and just this muscle, or like this muscle hurts. Well, that's because only that muscle has the receptor that the virus can bind. And so only that muscle or neuron or eyeball can get sick, right? And so, for example, with HIV, some people can get HIV in their nervous system. Most people get HIV in their immune cell population. And so it just depends on the match between the host and the pathogen. So that's one factor. Another factor is your overall ability to respond, right? What's your immune system? Are you compromised? Have you seen this pathogen before? We're going to do an immune lecture in a couple weeks. When we do that, you'll get a chance to see, like, how's the body's immune system respond. So some of it is just how nasty the virus is, how many viral particles it's able to make. Some of it is which cell types, and some of it is your personal makeup. Okay. So epigenetics is this cool new thing which helps us understand a lot of how things can change when our genes aren't different. So if we look at identical twins will sometimes have really different gene expression profiles, by which I mean they may turn on or off different genes. They might have, they're not even though genetically they're identical at the level of this gene's nucleotide sequence, they're not exactly the same. And they're not exactly the same because different genes can be turned on or off based on structural changes to the DNA, not whether it's ATGC, but other things. And we're going to look at those in just a second. Those structural changes can be inherited. So some things are really problematic. For example, Agent Orange, which people were exposed to during the Vietnam War, in Vietnam, turns out to cause epigenetic changes in the individuals exposed. Those changes to the way the DNA is organized is inherited such that we see those changes in the grandchildren of the soldiers who came back from Vietnam. And those genetic changes they're not ATGC changes. They're structural changes to the DNA. They seem to be associated with a series of current diseases. There's a study that suggests they may be involved in increased rates in autism. There's another study that suggests they're generating an increased susceptibility to certain kinds of cancer. This is probably also the case for many other environmental toxins. Stress. seems to do this as well. So there's a researcher on our campus here that is looking at the impact of stress during pregnancy and how that stress on the mother changes the epigenetic characteristics of the fetus. For example, specifically the moment she's looking at women who were pregnant around the time of 9/11 and how the stress of that incident changes the genetic organization of their babies and how that affects their learning and their long-term outcome. So what are they? There are two types of alterations. One is what's called DNA methylation, and the second is chromosomal reorganization. So methylation, if you're taking your chemistry, some of you I know have or are, methylation is adding a methyl group, which is a CH3. You can see that here. So this is not methylated cysteine, and this is methylated cysteine. It just adds this methylation to the DNA. And the methylation almost always occurs on a C that is next to a G. So these are called CPG islands. So you'll have a CG, 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 and in this region, you will have methylations added. And these methylations tend to occur in areas that are promoters for genes, and they tend to turn genes off. Okay? So this is repressing a gene, not with a protein, but with an actual change to the DNA. And these are inherited changes. Yeah?
So these, these often occur in genes that are promoters?
So these occur in CG-rich regions in promoters. Okay, and so we can inherit these changes because when you have a methylation on one side, you can have that methylation also placed so it's on the parental strand, and because it's on the parent strand, when you do the replication, it's added to the newly produced daughter strand as well, and therefore it can be multi-generational. So you need special enzymes called demethylations to remove them. There are some drugs now that are being tested actually all the way into humans to see if we can remove some of these methylations in a way that improves life outcome for individuals with certain diseases associated with this kind of problem. So methylating the DNA tends to attract proteins that are involved in repression and tends to inactivate DNA. This is important, especially in embryonic development. One of the things that happens during embryonic development is we need to turn a lot of genes off, and then we need to turn a lot of genes on and then slowly turn them off. So in a developing embryo, we have a lot of demethylases, a lot of enzymes that demethylate and then allow these genes to be turned on. Then we don't need them once you're grown up, we turn them off. There are also often signs of misregulation in cancer. So in cancer, a lot of genes get turned on and off inappropriately, and that can be because the methylation is incorrect. So sometimes they'll take a tumor and they'll analyze its epigenetics, its methylation, and that can help them figure out what's the best medication to treat your particular cancer. Yeah?
Could methylization create conjoined twins?
So methylization is not playing a role in conjoined twins. Conjoined twins tend to occur when an embryo is about to divide, when the cells of an embryo divide to make two, but they don't completely separate. And sometimes that just happens because the cells are not sticking together in the way that they should. We're going to have a lecture on development, but there's a phase in development called compaction when you have a bunch of individual cells that compact to make one embryo. If that doesn't occur correctly, and they will separate and make two, and sometimes they half separate. Another reason it can happen is that there's actually a mutation in a few cells, and those few cells are trying to be pushed out of the embryo because they have a different stickiness, a different adhesion, and then they don't make it all the way out. So conjoined twins are not related to methylation. We can also have remodeling of the chromatin. So again, you should have learned about histones. Their DNA wraps around the histones to make the chromosomes, and then they pack tighter and tighter and tighter to make that chromosome structure that we see. We can acetylate those histones or deacetylate the histones. Acetylated histones are going to be open. If the DNA is accessible, we're able to get in there and turn the genes on. If the DNA has histones that are deacetylated, then they're packed really tightly, and that prevents the DNA from being accessible. These can, and I mentioned some of this, right? So environments can cause changes to the epigenetics that can be inherited. Twins can have different exposures that will generate different things. Stress can generate it. Addiction and psychoses can also generate these issues. These can be inherited, and they're usually inherited differently depending on whether the mother was exposed to the stress or the father was exposed to the stress. So it turns out that you, for the most part, inherit your mother's DNA methylation patterns. So in the egg, the embryos, in the egg, the DNA, the chromosomes there, tend to maintain their methylation, whereas in the sperm that are produced, the methylation is removed. And this is to help prevent competition between which is the right methylation state for the developing embryo. If mom's chromosomes are one way and dad's chromosomes are another way, they may be in conflict. So mom gets to keep hers, dad loses his, and together then you end up with an embryo which has mom's epigenetic memory. Which is not to say that sometimes the dads are inherited too. We can identify when these chromosomes are acetylated or non-acetylated, when the histones are acetylated or non-acetylated, because the chromosomes pack really differently. So here is a cell where you can see these bright yellow spots. The bright yellow spots are DNA that is really tightly packed in these deacetylated histones. And in this case, this is the X chromosome. So you may know from previous lectures that females have 2 X chromosomes, males have 1. In female cells, one of those X chromosomes is inactivated because we need a balanced number of them. And in mammals, we turn off one of them, and so they get turned off by being wrapped up super tight. And we can see that here in the cell. Yep.
For this slide before this, can certain traits be?
That's right. So certain traits will be differentially expressed in the resulting offspring based on whether the allele is inherited from the mother or inherited from the father. So there's some funky conditions. For example, there's a condition called Prader-Willis syndrome, which is an epigenetic inherited disease when you get it from your father. If that exact same gene is inherited from your mother, you get a completely different disease. It's called Angelman syndrome, and the phenotypes are completely different. So there are definitely circumstances where, depending on who you inherit what from, because of this methylation situation and this histone packaging situation, you can have completely different consequences. Yeah, so Angelman syndrome and Prader-Willi syndrome, same gene, one's inherited from the mom, one's inherited from the dad.
Can it prove to be beneficial at sometimes?
Can it do what?
Can it prove to be beneficial at
Can it prove to be beneficial? Sure, everything can prove to be beneficial. I'm dyslexic. It makes me really awesome at my job because when I read, everything flips upside down and backwards, and I study how cells move in three-dimensional space, so I can close my eyes and everything flips around in three-dimensional space, and that's awesome because that's what I need to do, right? So I'm sure there are circumstances where it's advantageous. Yeah. Okay, so X chromosome inactivation, is that it? Which thing? I just want to make sure I don't miss anything. Oh yes, we need to do alternate splicing. Okay, so someone asked earlier, why do we have these introns? One of the reasons that we have introns is that it allows cells to be put together in slightly different pieces. So here we have a single gene in a eukaryote. This gene has many exons and many introns. We make a messenger RNA that includes all the exons and introns, and then we use alternate splicing to put them together in a different order. So if you look, this first protein is made from the mature messenger RNA that it contains exon 1, 2, 4, 5, and 6, whereas this one next to it contains 1, 3, 5, and 6, and the one after it contains 1, 3, 4, 5, and 6. And so you may ask, well, why is it happening? Why are we doing this? There are several reasons. One is that it's an easy way to make related proteins that are slightly different. So for example, we have myosins that are expressed in lots of different places in lots of different ways, different times in development. And rather than having 15 separate myosin genes. And actually, I think we have more than that. We have a few less because we can organize them a little bit differently using this feature. Now, there was a question. Did I answer it? What was your question? Someone? No, it was, okay. I'll come back to you if you still have one. I thought I missed a hand in my conversation. So that's one of the reasons why these types of proteins can be valuable. And we'll look in other lectures as to some other examples of specific cases where the properties of the proteins change and how that can be helpful. You have another question?
So you see how in the gene,
Yep.
there's only one of each exon?
That's right.
How in all of the bottom are there?
Oh, so this is just different examples, right? So this messenger RNA could go option 1. It could go option 2. It's not going to happen in an alternative way. All right, I am going to wrap it up. I would love it if I got some feedback from how this video recording thing worked on your end, because if it didn't work, I need to know so I can fix it before we have to use it. Comment? Question? So it automatically shows up here. So remember I told you I was going to say lecture 1213? Right here, it should, as soon as it's finished processing, have that go for you.
Thank you, Dr. Holtzman.
You're
I will give
welcome.
you emails and I
Please
get
let
feedback
me
for
see,
it.
like,
Yes.
did I just delete the first half by making the second half? Like, I don't know what happened. So
Thank you so much.
you're welcome.
My name is Abdullah. I wanted to ask you some questions about this. Like, exons that are removed when you're splicing?
The exons get put together and the introns are...
Introns are spliced. Okay. And that happens after all of the
I got it for a while.
That happens to take an immature messenger RNA to make a mature messenger RNA.
When this happens, you can just take any one of the exons and make them into different patterns? They make them...
So generally there's a logic behind it and there's a specific... Each cell will make a specific one. It's not random. So there are regulatory processes that say, oh, you're this cell type at this point in embryonic development, make version 2. Oh, you're that? Okay, version 6. So a few cells will make 2 or 3 options, but most cells will pick one and make only that one. So
Depending on what you need?
Depending on what you need, which is defined by, right, so I always love these words, like what does the cell need? How does it know? Well, it knows because there are a host of these little proteins that bind the sequences at the breakpoints between each one's exons.
And these exons aren't necessarily one codon, right?
No, they're usually much more. They can be, 10 to 1,000.
All right.
They can, yeah.
All right, so does that mean there's only one stop codon or something there?
So sometimes there'll be 6 different exons that contain a stop codon, and so that allows you, because you need a stop codon at the end of your mRNA, right? So if you either need a circumstance where codon 10 contains a stop codon and every single resulting messenger RNA contains codon 10, or you need 3 different exons that contain a stop codon. But